Hello, good evening everyone. In today's class, I will discuss a very important topic which is blood coagulation. Okay, we all know that blood coagulation is very important to prevent blood loss, right? Before going to this topic, you need some details, some very basic things like here I have reference of the structure of a artery because the blood flows through a artery. Okay, and you can see here that it has mainly three layers. What are the main three layers? It's tunica intima, tunica media and tunica externa. What are these three layers? Basically tunica intima, which is made up of these primary cells that are endothelium. Okay, the cells are present here, they are known as endothelial cells, and these are very important cells. The wall of the capillary is made up of endothelial cells. Okay, after these endothelial cells there is another layer, that is tunica media. Now why this tunica media is important? Because of the stretchability of the artery. Like we have to stretch the artery, we have to be flexible because of the maintenance of the blood flow. So here is the presence of another tissue, that is smooth muscle. Because of smooth muscle these arteries can contract and relax. Okay, and after that there is another fibrous layer, that is tunica externa, and which is member of external elastic membrane. Till this you can understand, hope you can understand. If you still have any kind of doubt, then please write it in the comment section. Okay, definitely I will explain it after some time. Actually, blood coagulation is a process of some sequential events. Okay, it's a part of some sequential events. What is that? For blood coagulation there are before blood coagulation there occurs another two steps, and this whole process, from the entire activation of platelets to the formation of blood clot, this whole phenomena is known as hemostasis. What is called? It's hemostasis. Okay, that means hemostasis means the breakdown of these molecules. Okay, so what happens? Firstly, it's vascular spasm. Vascular spasm means it's like a tonicity that we feel after getting any cuts. Okay, so this is called vascular, means the whole blood vessel systems. Okay, it gets damaged, and because of its damage we feel pain, so that is the vascular spasm. Okay, and then the next one is the platelet plaque formation. That means whatever I am trying to say, it's if it is a structure of our blood vessel, and this is the endothelial cell lining that I have previously said and show you in the diagram of a blood vessel. So blood vessel is made up of endothelial cell layer. Okay, so this is a picture of blood vessel. If it is a artery or it's a vein, whatever it is, the blood is flowing within this capillary, blood capillary, and there is with plasma. Here is the another blood cells, like the majorly seen the RBCs, the red blood cells. Okay, this red color are the RBCs. Then there are some other elements like WBCs, they are also circulating with the blood. And there is the another molecule whose function I am discussing, that are platelets, and these platelets are responsible for the coagulation of blood. Now this is a intact capillary. So if there is any breakage, that means that endothelial cell layer will not be continue, they will break. So what happens if there is any breakage? So if there is any breakage, like these endothelial cells get ruptured, there is a cut in the capillary and blood is trying to oozes out. Okay, and this, after this capillary layer, there is another layer, that is a collagen fiber. Okay, this layer is the collagen fiber, right? It's a collagen fiber layer, okay. And under this collagen fiber layer, there is another layer that is known as the smooth muscle layer. Okay, so there is a cut and the blood is comes out within this, from this blood vessel to the outside of the capillary, and we need to prevent this blood loss. So for preventing this blood loss, we have to activate these platelet molecules. What are the platelets? These are the platelet molecule. We have to activate them because they are circulating in the blood, they are not in the site of infection. We have to call them to the site of infections. Where is the site of infection? This place. So this molecule has to come to this place and they have to aggregate in this place. Okay, because we don't have any gum to replace or to prevent this blood clot. We cannot put any gum here, right? So we have these natural molecules that are platelets, and you can see they are very small in structure. So only one platelet is not able to prevent this whole blood loss. That's why we need more in numbers. So we have to call so many platelets to the site of injury, right? So this all process, that I'm just giving you a brief idea that how it will look like, because I will not discuss all these 5 steps in this video. I will only describe the blood coagulation, that so that you can have a brief idea that why I'm going to discuss blood coagulation. That's why I'm just giving you this brief sketch. So if I draw this platelet molecule in a bigger size, and they will change their shape. Okay, so the platelet, it's look like this way, they change their shape like this way. This is the modification, and they attach in the site of infection, because you can see this, there is a blood loss. Okay, so they are sit like this way, and there is so many platelets get aggregated. So this is called the platelet aggregation, and for this step to happens, there is so many other molecules that play a very crucial role for the activation of platelets. Okay, and the another thing, so it's form a plug, it's aggregated. Okay, so because we don't have cotton in our body, so this platelet plug, naturally whenever we have get any kind of injury or clot, we put cotton in that cut area, right? So we put platelet like aggregated form, so that it can act as a plug. So it is known as platelet plug formation. It is known as platelet plug formation. platelet plug formation. But I will not discuss all this process and my video will get larger. So if you are interested to know this whole process, you can write it in the comment section. Definitely I will make a separate video for this whole process. Okay, so if we consider that our platelet plug is formed in the site of injury, then what happens after that? That will be the discussion of blood coagulation. Okay, so we will discuss now blood coagulation. Now comes to the blood coagulation. And this blood coagulation pathway occurs by following the two other pathways, that are intrinsic pathway and extrinsic pathway. Okay, so blood coagulation is split into two common pathways, that are intrinsic pathway and extrinsic pathway. And you can see that this intrinsic pathway and extrinsic pathway are combining and they are producing the final common pathway, right? So what is this type of pathways? There are activation of factors which will lead to formation of blood clot. Okay, so for the activation of blood clotting factors, we need this type of pathways. There are 13 factors. How many? There are 13 factors. Okay, so we will see the activation of these 13 factors by the means of intrinsic pathway as well as extrinsic pathway. Okay, and there are the names of the factors. So firstly we will see the pathway, intrinsic pathway, and I would like to see a tricks to remember these two pathway. This is not a complicated pathway. Many times many student thinks that this is a very complex pathway, but it's very simple. See how you have to remember. Like there are 13 factors, so there will be a chain of events. For the activation of factor 1, we need to activate other 12 factors. Okay, so factor 1, which is fibrin, which will form the clot. Before activating factor 1, there are chain of events, and that is why this is known as a cascade. Cascade means chain of events, chain of reaction, which will not stop till then the end product will be produced. Okay, so what happens, there will be activation of 13 factors. How they will activate one by one, we will see through this two pathway. So for intrinsic pathway of blood coagulation, okay, for intrinsic pathway of blood coagulation, there will be firstly the factor 12 will be activated. And this I would like to say one thing, that these all factors firstly remain in inactivated state. We have to activate this all these factors because of the blood loss, because we need to prevent the blood loss. Okay, so they are all in inactivated state. So factor 12 is followed by the activation of factor 11, and then the tricks is that there will be no 10. So 10 is not, that's why we will not write 10 here. So after 12, then 11, then 9. Okay, after that 8, and then 10. There will be the activation of factor 10. And for intrinsic pathway, they will be activated as there will be the factor firstly is the factor 3, and then 7. So factor 3 plus 7, it's 10. So this will be the activation of extrinsic pathway. It's extrinsic and it's intrinsic. Okay, so for intrinsic pathway there will be activation of 12, 11, 9, 8, 10, and here 3 plus 7, it's 10. And then the final common pathway. For final common pathway, there will be the activation of its multiples like 5 and 2. Then there will be the activation of 1, which is fibrine. Okay, and the clot will be formed. So see, there is the intrinsic pathway. Intrinsic means that it occurs within the blood vessel. So whenever there is a cut in the endothelial cell layer, the factor 12 is get activated by, or whenever it get in contact with collagen. Okay, because we know that after endothelial cell layer there is a connective tissue layer, that is collagen in the blood vessel structure. So that is why factor 12, okay, factor 12 which is in inactivated state, inactivated factor 12 will become activated. Now it's factor 12A means activation. A means activation. Now it's inactivated factor 12 gets converted to activated factor 12A when it gets contact with collagen. So activation of factor 12A occurs whenever there is damage in blood cells or the blood cells in contact with collagen. Okay, that is why it is known as intrinsic pathway, because it occurs within the blood vessel. There is no other tissue, no other external molecule is involved in the activation of these factors. These factors is getting activated by its own contact, that is collagen. Okay, now there comes, I have told you that this will be a chain of reaction. So now after factor 12A, this factor 12A will help in the activation of factor 11. So factor 11 now getting converted to factor 11A. So this is the inactivated factor 11. This is inactivated in condition. Inactivated factor 11A will be converted into activated factor 12A in presence of this blue arrow, means it helps, it need the for its activation. 11A or factor 11 needs the help of factor 12A and also these two molecules that are pre-kallikrein. I repeat, it's pre-kallikrein. Okay, it's the pre-form of kallikrein molecule, which is a protein, and that after that pre-kallikrein will produce that kallikrein. It's activated from its kallikrein, and this kallikrein will produce that kininogen, and this kininogen will produce bradykinin. Okay, maybe after some time or on a separate video I will let you know what is the function of these molecules. Okay, but this pre-kallikrein to kallikrein and kininogen, these molecules, these are also present at the blood vessel, and they help for the activation of factor 11A. Okay, now factor 9. I have told you that after 12, 11 and then 9, not 10. So inactivated factor 9 will be activated factor 9 with the help of activated factor 11 and calcium. Activated factor 11 and calcium. Okay. Next, there will be, in the next step, there will be the activation of factor 10, and this is very interesting. Till then, if I consider the activation of factor 12A is the first step of our reaction, this one is the first step, and the activation of factor 11 is second step, activation of factor 9 is the third step, and activation of factor 10 is the fifth step. Then there is one step is missing. What will be that fourth step? See, that fourth one will be activation of factor 8. Factor 8 to factor 8A. Factor 8A. Okay, factor 8A. And I will like, it's very interesting that know you that facts. I'd like to know you that fact that for activation of factor 10A we need a complex, and that is formed by factor 9 and factor 8. So factor 8A. Okay, so what we need for the activation of factor 10, it's factor 8A and factor 9A. So factor 8A plus factor 9A, it forms teenase factor, and that will help in the activation or conversion of inactivated factor 10 to activated factor 10A. Okay, is it clear? I repeat, there will be a combination of 3 plus 4 step. 3 plus 4. Activation of factor 9 and activation of factor 8. They will combinedly activate factor 10A in presence of the molecule calcium. Calcium is very important, and it is known as factor 4. Factor 4 is calcium, and this factor is very important for blood coagulation, but coagulation cannot take place if there is no calcium. Okay, next, after activation of factor 10A, what will happen? There will be activation of, I would like to say that for this pathway, intrinsic and extrinsic, commonly we will activate factor 2, which is thrombin. Factor 2, which is thrombin. And we need the final pathway to produce fibrin. So till then we will activate factor 2. So after activation of after activation of factor 10A, there will be the activation of its multiple. Remember the tricks I have told you, that here after 10 we need to activate factor 5 and factor 2. See, there is factor 2. For activation of factor 2, we have to activate its primary components like prothrombin activator complex. Okay, from factor 10A in presence of calcium and another factor, that is factor 5A. Here is the activation of factor 5 to 5A, and again factor 5A and factor 10A in presence of calcium, they will activate prothrombin activator complex. Okay, from prothrombin activator complex there will be generation of prothrombin. See, thrombin before thrombin it comes prothrombin. Okay, thrombin and its inactivated form is prothrombin. We always, there is always, the factor remains in always inactivated condition. Whenever we require to activate them, we activate them. Okay, so there is the prothrombin. So from prothrombin it's thrombin, and prothrombin is also getting activated by prothrombin activator complex. So firstly we need to provide or produce prothrombin activator complex, which is getting produced by the help of two factor, that is factor 10A and factor 5A. That's why I have drawn this arrow like this way. Factor 5A we required for the activation of prothrombin activator complex. Factor 5A and factor 10A. Commonly in presence of calcium they will activate prothrombin activator complex for the activation of prothrombin to thrombin, and this thrombin helps in the conversion of factor 5 to 5A. And the interesting thing is that this thrombin also help in the conversion of factor 8 to factor 8A. This is also thrombin. Okay, so I repeat, firstly factor 12, factor 12 to 12A, it needs to contact with collagen. Okay, then factor 11, it helps or it need the helps of factor 12A. Prekallikrein and kininogen, they are a type of protein molecule and they help in the production of bradykinin. Okay, next factor 11A and calcium, they both help in the activation of factor 9A. Factor 9A and factor 8A, they combines, and in presence of calcium they activate factor 10A. After that factor 10A in presence of factor 5A, they activate prothrombin activator complex, also in presence of calcium. Factor 10A, factor 5A and calcium, they combinedly produce prothrombin activator complex, which will lead to prothrombin, and from prothrombin to thrombin, this is the activation of factor 2 and completion of intrinsic blood coagulation pathway. After this flowchart, you have to write the name of the figure. Don't forget. Okay. Hope you have understood. Next, the intrinsic blood extrinsic blood coagulation pathway. This is called extrinsic as it occurs outside of the blood vessel in presence of any tissue factor. Tissue is different, so it will secrete some factors and that will help in the activation of the factor 3 and factor 7. So whenever there is ruptured of blood vessel or damaged extravascular tissues, these tissues release the factor tissue thromboplastin, and it is the factor 3. It is the factor 3 which is tissue thromboplastin, factor 3. Next, factor 3 is basically a compound of lipoprotein, that means lipid with protein, and phospholipid molecule. Its composition is lipoprotein and phospholipids. So factor 3, whenever it gets activated, it will activate the factor 7. So inactive factor 7 get activate in presence of factor 3. Okay, next step is the activation of 3 plus 7 is equal to 10. So next step is activation of factor 10. So factor 10 is getting activated to its for activated form, factor 10A getting converted. Inactive factor 10, it's getting converted into factor 10A in presence of factor 7, and factor 3, which is TTH, means tissue thromboplastin, and PP means phospholipids and calcium. Okay, so it is the factor 3 and calcium. Factor 7A, factor 3A and calcium, they combinedly activate factor 10A. And after that there will be the common step, that is that I have already discussed, that from factor 10A we have to produce PAC, that is prothrombin activator complex. It will be activated by two factors, factor 10A in combination with factor 5A and calcium. From prothrombin activator complex, there will be activation of prothrombin, and from prothrombin to thrombin, and then this thrombin will produce or help in the conversion of factor 5 to factor A, so that this production of factor 5A cannot be limited, so that whenever we process the blood coagulation, it remains available. We can get 5A easily, so that thrombin helps its uptake. Okay, so factor 5 to 5A. Next, so it is the extrinsic pathway of blood coagulation, and it's very simple. Okay, only you have to remember the activation of factor 3 and factor 7, because factor 3 and factor 7 combinedly in presence of calcium activate the factor 10A, and after the step, that is activation of factor 2, that is common with intrinsic blood coagulation pathway, that is same as intrinsic blood coagulation pathway. But this is not the least, because we have to produce fibrin. Till then we have produced or activate factor 2, that is thrombin, but there is a final common pathway which lead to blood clot. See, this is prothrombin. Okay, so you remember that prothrombin is activated by prothrombin activator complex. So the before this, that is the prothrombin activator complex, which is activated by factor 10A and factor 5A and calcium. Okay, so from prothrombin activator complex, there is prothrombin and there is thrombin, that is factor 2. So factor 2 is activated. Now this thrombin, okay, this thrombin, what causes the thrombin produce fibrinase. This thrombin produce fibrinase, which is a enzyme. Okay, which is a enzyme. It produce fibrinase. What it produce? It produce fibrinase. Fibrinase. It's a enzyme. Okay, so thrombin helps in the activation of fibrinogen to fibrin. Fibrinogen to fibrin. Fibrin, its inactivated form is fibrinogen. It forms, thrombin helps in the formation of fibrin. Okay, from fibrinogen, and it also produces fibrinase enzyme. Okay, so what is the role of fibrinase enzyme? It forms the clot. It forms or catalyze the fibrin to fibrin polymer. So here is the role of fibrinase. They catalyze and form a fibrin polymer. Okay, and there is also the activation of, factor 13. Factor 13 to 13A, because factor 13 is known as fibrin stabilizing factor. This is known as fibrin stabilizing stabilizing fibrin stabilizing factor. Okay, so there is formation of fibrin monomer. Monomer means only one unit, but we need fibrin called polymer to form a clot. Only one molecule is not enough to prevent the blood loss. That's why we need a lot of fibrin. So there will be a lot of fibrin. Okay, and lot of fibrin will be attached with a bond like this way, and this bond is created by this enzyme fibrinase. That's why it helps in the formation of fibrin polymer. So there will be a like this way, this fibrin, this fibrin, okay, this fibrin, and they are like this way. They form a net like structure so that they can be placed over the platelet plug, and it will prevent the blood loss that will form a clot. Okay, so, and I missed that factor 13 will be activated with the help of thrombin and calcium, and it will be converted to factor 13A. And to stabilize this clot, we need factor 13A. So, what happens here? Prothrombin to thrombin, thrombin leads to the activation of factor 13 to 13A in presence of calcium. Again, thrombin helps in the conversion of fibrinogen to fibrin, and also it produces fibrinase enzyme. What happens? Fibrin to fibrin polymer, and this bond is stabilized or formed by the enzyme fibrinase, and this formation of this fibrin clot or polymer will be stabilized by the fibrin stabilizing factor, that is factor 13A and calcium. Okay, hope you have a brief sketch of blood coagulation. Now what you need to do is practice. Just write down these easy steps of blood coagulation for at least 5 times, and then just remember or try to recall. See the magic. Okay, so stay tuned with me. Good night. Bye bye.